Welcome, everyone, to the Emerging Therapeutics and Neuroscience panel. At this time, I am honored to introduce Dr. Robert Pacifici, Chief Scientific Officer of the Cure Huntington's Disease Foundation, and also UCLA Technology Development Corporation Board of Directors as the moderator of this panel. And at this point, I'd like to turn over to Robert.
Well, thanks very much, Mark. It's a privilege to be here hosting this session. I hope the title of our session is not an oxymoron and that there are indeed emerging therapeutics in neuroscience. Everybody knows that drug discovery is a tough business, and I would say that trying to discover drugs for diseases that affect the brain is definitely the advanced class. We've got a huge challenge. But that means that there's also a huge unmet need and a huge scientific and economic opportunity for those of us that are in this space. And so I'm delighted to welcome a very distinguished panel of participants that come to this problem with a variety of viewpoints, a variety of expertise, and from different sectors. So what I'd like to do is start off by asking each one of the panelists to do a brief introduction, your name, your position, the institution you're with, and your areas of interest and expertise. And then I'll tell the audience that we're more than happy to entertain your questions. I'll be looking at my screen to see as they come in. But we also have a collection of topics that we've pre-discussed, and we'll see how those overlay with the interests of those of you who are dialing in to listen to this panel. So without further ado, I'll start with the local team advantage of Dr. Dan Geschwind. Maybe I can ask you to lead us off, Dan. And unmute yourself as you come on. There you go.
Good point. Thank you. So thank you, Robert. And I just want to say it's a great pleasure to be here and participate in this panel. I want to thank Mark Wisniewski for organizing this along with Robert, as well as the other panelists in our preparatory discussions. So my name is Dan Geschwind. I am the Gordon and Virginia MacDonald Distinguished Professor of Neurology, Psychiatry, and Human Genetics at UCLA. My roles in those departments include directing a neurogenetics program, as well as a Center for Autism Research. And institution-wide, I'm the Associate Vice Chancellor and Senior Associate Dean of Precision Health and the Director of our emerging Institute of Precision Health. So I thought I'll just spend a minute or two just to introduce our laboratories research and most of what my research piece of the career has been about, which focuses on two areas of large unmet need in brain diseases, autism and neurodevelopmental diseases, and neurodegenerative dementias, across two things across the lifespan. And I have three guiding principles that I'll share with you, and then let others introduce themselves. But that I think might be worth talking about. One of them is leveraging the power of genetics and genomics to take an unbiased and more holistic view of disease. The second has been the prudent use of model systems with a clear consideration for evolution. And this is especially relevant to mouse models. And the third, another important topic, is to consider disease heterogeneity. Our clinically defined disorders are heterogeneous. One size doesn't fit all. This is where genetics and precision health, big data, and discovery really comes in. In fact, I'm not going to focus on precision health here, but we're having a lounge at 2 p.m. on that. With regard to genetics and genomics, the key is to start with genetic risk, which is a causal anchor, generate large data sets, and subsequently integrate these using systems biology and network methods. And then, of course, finally, testing any models that you have experimentally. And, this has been a very powerful model that we've been using literally for the last 20 years. And I think it's finally coming into more relief and being more widely used. So again, the genetics and genomics, the evolution, the judicious use of model systems, considering evolution.
Great. Thanks, Dan. Those are themes we'll definitely pick up on during the discussion. Linda, maybe we can ask you to go next.
Yes, and thank you, Robert, for inviting me to join this panel. My name is Linda Liau, and I am a neurosurgeon. I'm chair of the Department of Neurosurgery here at UCLA, and I also am director of the UCLA Brain Cancer SPORE, which is an NCI-funded program, and SPORE stands for Specialized Program of Research Excellence. And the crux of my research has focused around brain tumors and brain cancer, but more specifically, I live in that translational realm where we're trying to take new therapies that are developed in the laboratory into first-in-human trials and early phase 1, phase 2 trials, and I think I concur with Dan's assessment about the need for precision medicine and genomics and how to integrate all that data into our developmental therapeutics. But also, I think there's there are pain points in terms of getting lab research, lab discoveries, into patients, and all the way from the early stages all the way to phase 2, phase 3, and then out into commercialization, which I think is hopefully something we can discuss today.
Fantastic. Thank you so much, Linda. Richard, maybe I can ask you to unmute and introduce yourself.
Yeah, hi, Robert, distinguished co-panelists. I'm really delighted to be here. I have a signed promise that if I participate in this, that the Lakers and LeBron James are going to win another championship, and that's the main reason I'm here. So my career started in Cleveland, and first about 40 years were in Cleveland. So I took full training in internal medicine, neurology, and then did a molecular biology postdoc for 5 years. I set up a lab and a clinical practice afterwards at the Cleveland Clinic and remained there for 25 years. At the Cleveland Clinic, I took care of people with multiple sclerosis, and I started researching neuroinflammation in the context of MS. As MS got more and more treatable, mainly by aiming at the inflammatory aspect of MS, it seemed that we could apply our learnings to other conditions. So I set up something called the Neuroinflammation Research Center, which was meant to be the one-stop shop for all of neuroinflammation research in the local area, and extended across all neurological indications. In 2014, I moved to Biogen to set up a neuroimmunology group there and spent 3 years doing that. And then 3 years ago, I moved to Third Rock Ventures. Third Rock Ventures is a venture capital firm that uses its resources specifically and only to set up new companies. So the activities that you carry out in the lab, your discoveries in the lab, as Linda mentioned so aptly, need to be translated, need to be brought into the clinic. And the specialty at Third Rock Ventures is to set up new biotech companies which are meant to endure and last. So we don't invest in single programs, we invest in what are called platform companies. In the 14 years of existence of Third Rock, 56 companies have been set up. Almost all are still in existence. 2/3 have gone either to IPO or acquisition, usually by a large pharma company. And most importantly, there are 14 products that have come out of a single company deploying less than $3 billion in 14 years. So that's an extraordinary sort of example of what can be done. And it involves a rather well-developed and sophisticated strategy for setting up new companies. And so glad to be here and talk about that now. I'm a venture partner now at Third Rock, which means I'm involved in setting up companies mostly around neurology and immunology, because that's what I know about. And I'm the presumptive CMO of a company that's going to launch in less than a month and come out of stealth you'll be hearing about. So
thanks so much, Richard. That's great. Maybe we can move to Greg next, if you can unmute and introduce yourself, please.
Okay, thank you, Robert, and thanks for having me here today. Hi, everyone. I'm Greg Rippon. I'm the Chief Medical Partner for Neurology at Genentech. Despite being at Genentech, I'm actually based in Southern California in Thousand Oaks area. And what we concentrate on is all of the medical affairs or medical activities in support of Genentech's marketed and pipeline products in neurology. That spans the realm of evidence generation activities, mainly bringing in the products into the post-marketing setting, real-world evidence generation, and investigator-initiated research programs and other avenues of evidence generation. In addition to a comprehensive medical communications programs in support of each of the company's programs, and engaging with the external community. So we have very significant engagements both with practicing clinicians, professional societies, as well as advocacy groups and patient groups. And in addition, importantly, generating evidence in order to support access for patients to Genentech's medicines. A little bit of my professional background: I'm a neurologist. I'm an aging and dementia subspecialist by training. I've been in industry for about 16 years in clinical development and medical affairs roles, initially with Merck and Cephalon. I was the medical affairs lead for the Alzheimer's immunotherapy portfolio at Pfizer. I also moved to California about 5 years ago to build the neuroscience medical team from the ground up at Amgen, and I was accountable for the medical strategy and bringing Arenimab or Aimovig in migraine prevention to the market. I've been at Genentech for about 1 year, and my scope is I lead the U.S. Medical Affairs organization for neuroscience.
Excellent. Thank you so much, Greg. I think we'll move to Viviana, if we can ask you to unmute and introduce yourself as well.
Thank you. It's an honor to join the panelists, and I hope I can contribute my academic perspective as a professor of neuroscience and biological engineering at Caltech. And I also direct the Molecular and Cellular Neuroscience Center of the Chen Institute at Caltech. And with my group and collaborators over the years, we've been focusing on protein engineering to develop methods for supporting neuroscience research and translation. And in particular, I've been developing opsins for optogenetics and gene delivery vectors by modifying the adeno-associated viral capsids. So cargo optogenetics delivery through viral vectors. And I have to say, this is a very exciting week because less than 15 years since we've done the first mouse optogenetic experiments in vivo with fiber optics and viral delivery and just a lot of troubleshooting early on, figuring out all of the details. Just less than 15 years, this week we learned about first-in-human success with visual restoration, partial, but it's a great start with optogenetics and AAV gene delivery. So it's a wonderful moment, and it's very suitable for emerging therapeutics because I think the ability to have high frequency control for neurons will be key to expand these modalities to circuit dysfunction. So I'm very excited about contributing that. I am a basic scientist. However, I'm very interested in translation, and I've been supporting my trainees as much as I can with that. And I'm also a co-founder and a board of directors member for Capsida Biotherapeutics, a company that just came out of stealth recently in Southern California.
Excellent. Thank you so much, Viviana. I appreciate it. So for those of us behind the scenes, there are a lot of names up on the screen. And let me just pause briefly before I introduce myself to make sure I haven't missed any panelists. I think we got everybody. So my name is Robert Pacifici. I'm a biochemist by training. I actually trained at the other university down the road, USC. but grew up professionally at Amgen, so it's a pleasure to see their sponsorship of this event in a big way. I also worked at Zencor, got to see my old boss Basil yesterday in one of the other panels, and then spent some time at Eli Lilly and Company. For the last 15 years, I've been the Chief Scientific Officer of CHDI, which is a nonprofit organization that is exclusively dedicated to accelerating therapies for Huntington's disease. Maybe I can start there with a couple of the themes that you heard from our panelists already. You know, we all recognize that genetics can be incredibly powerful. In fact, as many of you know, Huntington's was the second gene ever positionally cloned related to a disease, fast follower to cystic fibrosis. And while it's clearly a monogenic disorder with 100% penetrance, in other words, everybody that gets the disease has the bad gene, everybody that has the bad gene eventually gets the disease. There are two things about that sort of represent a double-edged sword. The first one, maybe relates to one of the bigger challenges that we have in this field, which is developing predictive animal models. Over the course of my tenure at CHDI, we've developed dictyostelium worms, flies, zebrafish, songbirds, mice, rats, sheep, pigs, and non-human primates that express the mutant Huntington gene. And I've never met an organism that's happier expressing that mutant gene than when it's not. I just have no idea what the relevance of those particular animals is to what happens in a big, wrinkly human brain over 40 years. So I think there's certainly a need and an opportunity to use preclinical species to inform our studies. But I wonder if I can ask, maybe I'll start with Richard, put you on the spot. Since there are now therapies for multiple sclerosis, there is the opportunity maybe to do some retrospection and look at the reverse translation of what's actually happened in the clinic relative to some of the animal models that might have been used along the way, and what some of the lessons learned from that might be.
Superb question, probably inadequate respondent, but I'm going to do my best. The animal model that's most widely used for multiple sclerosis, of course, is experimental autoimmune encephalomyelitis, called EAE. It's the oldest animal model of disease, and it was discovered serendipitously through research into what was called the neuroparalytic catastrophe that occasionally occurred in people receiving early versions of the rabies vaccine. So in the 20s and 30s, the question was diced into two sort of broad categories. Was it residual rabies vaccine that was causing rabies infection, or was it something in the inoculum that wasn't the rabies vaccine, and the inoculum was prepared in duck embryonic brain tissue? So it's relatively simple to look at the pathology of people that suffered with this complication, which was about 1% of people getting it, and to see that they had a pathology that did not resemble in any way rabies, and the clinical syndrome didn't resemble rabies. And the next thing was to inject emulsions of duck brain into non-human primates and to obtain an inflammatory reaction that eventually became EAE. And over the many years, this was refined to a highly sophisticated model, which could be caused by T-cell clones that had a known T-cell receptor, a known MHC restriction element, a known peptide that bound the MHC restriction element and caused the disease. And for quite a few years, this was called essentially animal MS. And you would see people writing review articles discussing MS/EAE as though they were essentially the same thing. And what turned out to be the case was that just as the model was discovered somewhat serendipitously, many of the early treatments of MS were also discovered serendipitously and had nothing to do with developing the model. The model became quite effective for discovering mechanisms of antigen-specific immune reactions in the mouse brain. But the mouse brain is not the human brain, and the mouse lifespan is not the human lifespan. There is a condition that somewhat resembles EAE that occurs in people called acute disseminated encephalomyelitis, which can be a reaction to a variety of things, most often an immunization or a relatively trivial viral infection. But EAE is not multiple sclerosis, and most of the drugs that are used for MS were discovered either serendipitously or sort of went around EAE and direct into human experimentation. And the MS therapeutic armamentarium, as many of you are aware, is incredibly effective. Genentech has played a large part recently in this evolution, and it's a great success story. It is notable, however, that many of the things that work in EAE make MS worse, and many of the things that work in MS don't work in EAE. So it's an example of the fact that the mouse is not the human, and even if you design the model as cleverly as you possibly can, it's hard to use these sorts of systems for drug development.
So Greg, I don't think you and I overlapped at Amgen. I was there early days when there was actually a very rich pipeline of potential emerging neurotherapeutics, GDNF, leptin, BDNF, GDNF, NT3. All of those gave some signal, some very robust signal in animal models. Sadly, I think at one point, tongue-in-cheek, we renamed BDNF Big Disappointment, No Future, because we were so disappointed that it didn't work out. So as you're on the receiving end of data from the preclinical folks, what types of hallmarks do you look for in terms of efficacy that really pique your interest and get you excited about the prospect of something actually being a real shot on goal when it reaches the only beasts that actually have these diseases, us humans?
Yeah, it's a great question, Robert. I mean, I think in that question just underscores the complexity of neurologic disease and neuroscience in general, right? So, it's not uncommon, for example, in the neurodegenerative space to see a very impressive signal via imaging or other biomarkers that in later phases of development translates to nothing in terms of clinical improvements or slowing of decline. So that's been kind of a classic challenge as we bring molecules from the lab into clinical development in later phases, right? So I think to underscore the challenges that Richard was outlining, we see these challenges in terms of translatability of effects seen in animal models, all the way from the pain space to the neurodegenerative space and other areas of neurology. I think one of the advances from the MS perspective has been the development, and this took time, of in vivo biomarkers and imaging biomarkers and others that then in the course of later phases of development translated relatively well to our ability to predict clinical effects, and that's something that other areas I think have been a bit a little bit earlier on the curve in getting to that point, but I'm very hopeful that that's where we're heading. But making that leap from the lab to mid-phase development, solid signal on a biomarker that then translates to eventual clinical benefit in phase 3 is quite a challenge.
Yeah. No doubt. I think the lessons that we've learned is that it's probably naive to try and recapitulate a disease in toto in an animal model, and that what we've really been trying to focus on are specific mechanisms of action that we think are relevant to the disease, and even better if we can track those in both directions. In other words, down to the molecular level in terms of what receptor ligand axis, what signaling pathway we're tickling, but also to try and have the connectivity to the symptomology of the disease. Again, swinging for the fences and thinking about developing cures is probably ambitious as opposed to treating clinically meaningful aspects of the disease. As many of you may know, Huntington's is a multi-domain disease where there's the movement disorder. It was originally called Huntington's chorea because of the involuntary dance-like movements. There's an aspect that is very complex in the neuropsychiatric domain, and also in the cognitive domain, but unlike Alzheimer's, it's a loss of executive function rather than memory. And interestingly enough, if you talk to patients, you might see them genuflecting and having these Tourette's-like movements, and you might think, oh, that must be terrible. That must really bother them. And actually, they don't even notice their movements anymore. What they're really worried about is losing their marbles. They're worried about the loss of executive function. And so I think, one of the things I maybe can ask Linda to comment on is, how do we make sure that as we contemplate right from the beginning our drug discovery strategies, that we've got the patient perspective in mind, that we think about the end game and what is it that we're actually trying to treat short of curing the whole disease?
Yeah, I would completely agree with that. I think, in the world of brain cancers, we've cured cancers in mice, 100 times, but we can't do it in humans because of all the complexities of the animal models and how that translates to humans. So I think, the goal of the animal models or any models really is really mechanistic, learning more about the mechanism and the biology, and then, how that plays into how we design clinical trials and therapies going forward. And then with that being said, I think on the other end of the spectrum, I completely agree. We need to take into consideration what our patients want, and what drives them to enroll in clinical trials, and also what kind of endpoints they want to see. Sometimes we focus too rigidly on, imaging endpoints or certain kind of objective endpoints. And for the patient, that's not really, what's important to them. It's really, things that are related to function and quality of life. And I just recall, for instance, in treatments for spinal cord injury, we actually in our department, we have some researchers that are doing very innovative work in that area. And when you ask patients what bothers them, spinal cord injured patients, what bothers them the most, you would think that it's the paralysis, being paralyzed and not being able to move. But actually, it's the incontinence. It's not being able to go out, to a social event or to your kid's, baseball game without, being able, without, control of, your functions. And so I think we really need to have patients involved in terms of what it is that we are studying, and also endpoints for clinical trials. Completely agree.
Okay, thank you. A very thoughtful answer. I wonder if I could tap into Dan and Viviana a bit. As you guys contemplate your research strategies, I guess there's a couple of things that I'm sure you struggle with. One is really understanding the very complex underlying pathophysiology for some of these diseases. I'm very fond of telling people, if you don't know what's broken, it's very difficult to fix it. And how you approach that, how you're able to, in sort of more traditional drug discovery parlance, do the target identification and target validation that really gets you excited about proposing a pharmacological intervention point. And similarly, the sort of flip side of that is, what is the best pharmacological intervention? As we think about treating a very privileged organ, the brain, tough to get to, thanks to the blood-brain barrier, tough to measure, things that are going on there. I don't think any of us would contemplate doing serial biopsies the way some folks in the periphery can get away with. And so, what are the types of modalities when we don't have the luxury of just relying on small molecules that the field should really be focused on? So maybe I can start with Viviana, and then we'll go back to Dan.
Sure, happy to. And I'm very fond of Parkinson's and as an example, because for a while I've looked at it as a circuit disorder from the perspective of deep brain stimulation that works extremely well, although we still don't fully understand how it works. And those early experiments using optogenetics to try to understand how deep brain stimulation works showed me that we are looking too late in the pathology and at a very focal point that doesn't even have to do with the origin of the disease. Deep brain stimulation is applied to the subthalamic nucleus. The disease is caused by dopaminergic neurons degenerating in a different area, substantia nigra pars compacta. And this had us look at earlier points. What is earlier in PD development before motor and cognitive decline? There's sleep disturbances. So then we looked at sleep circuits. And even before that, there's gastric malfunction and transit problems. So this informed a route where and I think it can generalize across indications. What's the earliest point we can look at for neurospectrum disorders? And that might not even have to do directly with the brain, because the brain in itself is challenging. Could be late in the progression of the disease, and it's also hard to access. So to answer your question, maybe from two paths. One is for the disorders where we can look earlier and look outside of the brain, we should do that, and we should do more of that, even looking at the peripheral nervous system, for example. And for the disorders where we do need to access the brain, to bypass access challenges. And I will take your example of Huntington as an example. It's extremely hard to get to medium spiny neurons in the striatum. So let's say for the brain, yes, we can do invasive surgery and we can deliver locally. The spread is not that large, so you need to do multiple injections that have poor coverage and heterogeneous. You could, for example, access some areas adequately with ICV or intracerebral ventricular injections. However, striatum is hard to get. It is hard to get coverage for disease-relevant areas with direct injections. And for this reason, we've been working as a community over a long time now to try to bypass the challenge of crossing the blood-brain barrier for gene delivery. And there is progress. There is progress in animal models. But as you mentioned, no animal is the human. And even more concerning, what we learn with our own work, no mouse strain is necessarily the same as another mouse strain. So even between mice, there's differences as there are between people. So I think crossing the challenge of the blood-brain barrier, first we need to move from animals to humans. And then even when we look at humans, the solution might be different depending on the genetic background, depending on ancestry. And it becomes very important to take into account this heterogeneity, the genetic heterogeneity, that I'm sure Dan will have much more insightful and detailed things to talk about.
Viviana. Thank you. Dan?
What an elegant pass-off, Viviana. Thank you. That was seamless. I do want to reflect, as you mentioned and others have, and I tried to allude to it, this issue of the animal models and studying one mouse strain. It's extraordinary. Why do things fail? Well, there are a lot of reasons. It's complicated. We can't get into everything here, but, we looked back at one point and saw that pretty much every study that had been done in Alzheimer's disease, mouse models, whatever that is, were essentially done in the same strain. They were not strain aware at all. And as Viviana alluded to, these mice have hundreds of recessive, they're super inbred. So they're not even a representation of a mouse. And so one of the things we just decided to do is what happens if we outcross our mice and use multiple strains and look at our disorder on those multiple strains? And we were able to, and just one small point there is that when we looked at things that happened in an individual strain, those were not observed in human. And in general, things that were observed across all the very divergent strains were much more likely to be observed in human. Again, just but people have been kind of lazy a little bit, I would say, and dogmatic about the way that they approach things. And I think, so that's just one little piece of things. I would even though go much further than what's been said to say that, people talk about, well, let's use the mouse to look at a mechanism. We're just looking at it's a genetically sensitized system for looking at a particular mechanism. But without knowing if that mechanism is conserved, you don't really even know what you're looking at. So again, I think that there's been a lot of, assumptions made. The notion of the mouse as a small human is kind of inbred into almost all studies that have been published. And that's a recipe for disaster, especially when things like glia that Richard works on and others, and that are very important in many diseases, actually are among the most diverged cells, between mouse and human. So I think that's something very important to consider. I also want to endorse what Viviana said about nucleic acid therapeutics. I think that's an incredibly interesting area. Of course, there are all kinds of delivery challenges, immunogenicity challenges, and other things that we can discuss. So of course, small molecule easier, but their success really opens up a lot of possibilities for disorders that until now have been entirely untreatable, both in terms of replacing something that's missing or turning off something that you don't want on. And I think the kind of viral engineering that Viviana and her colleagues are doing, and as well as what's going on in industry in this area as well, really is quite promising in that regard. And the fact that these have been shown to be doable in humans in a few nervous system disorders, albeit mostly peripheral nervous system disorders so far, like at some spinal muscular atrophy and Duchenne's, and still is very, very encouraging, and kind of, I think, opens up a lot of windows too. So
thank you, Dan. So we have a few minutes left, and I wonder if we can go quickly around the panel. As I started off in the beginning saying, this is really challenging, the area of neuroscience, developing, therapies, and, perhaps no area has been as extensively studied and as extensively invested in as Alzheimer's. And yet, despite the demographers jumping up and down, yelling and screaming that this is only going to get worse, we still really don't have any effective therapies. So I wonder, if we could just be sober for a moment and maybe even self-deprecating and say, what are some bits of dogma that we really just need to flush down the toilet and get out of the way because they're not serving us well? I had the privilege of hosting Steve Paul as the keynote speaker at my conference a couple of years ago, and dyed-in-the-wool beta secretase guy said, maybe it's time to look at other mechanisms. So there's definitely dogma that we need to purge. And conversely, where, again, learning from Alzheimer's, and it took them a while. Where are there opportunities for something new, some collaborative effort, ADNI coming together to share biomarkers as pre-competitive space, thinking about dumping all of your clinical data into a common area so that everybody can learn from these failures instead of staying in competitive mode. So Richard, maybe I can start back with you, some dogma that we don't need anymore, and a new idea that might help melt this difficult problem.
Yeah, so I'm a general opponent of certain dogmas. One that I have been particularly troubled by is the concept years ago that there are good and bad states of microglia. These were called M1 and M2 microglia. And this was a reasoning by analogy from peripheral macrophages. Peripheral macrophages can be in an M1 interferon gamma dependent or M2 IL-4, IL-13 dependent state, but microglia don't. Microglia have things to do that peripheral macrophages don't do. And with the fullness of time and with the application of really excellent tools such as those deployed by Dan, such as single-cell RNA sequencing, it turned out you never see an M1 or an M2 microglial cell in vivo. You can create them in vitro if you wish, but they're not useful for anything. We're now seeing the same evolution for the astrocyte. So there's a proposal of an A1 and an A2 astrocyte, a toxic bad astrocyte and a tonic good astrocyte. And it's the same, for me, it's deja vu all over again. It's the same nonsense that we went through with microglia, and it will come to the same end. The problem with it is that people new to the area are going to read these high profile papers because these are easy experiments to do and easy to publish. And they're going to think, yeah, this is probably right. I mean, people who come into a new area, they're scholarly. They read, and then they work from what they read. And so for me, this has been a particularly troubling area. On the good side, I just want to highlight things that you, Robert, Dan, Viviana, Linda, Greg have already pointed out. One is that pre-competitive consortia in the AD field are beginning to make a difference. And so ADNI makes an enormous difference. The Alzheimer's disease sequencing project makes an enormous difference. And the general tendency to have to share data such that when you see a new paper come out with a good data set, it used to be the paper was the whole thing. Now it's the data set. I mean, it's nice they do an intro and a discussion and give you some references. That's not what you want. You want to dig into the data and have a good time and find something. So I find that extremely exciting. And I just want to recall that a disease that you mentioned, cystic fibrosis, took 20 years to get from the gene to the very, very, very first hint of an effective treatment. And so we're just at the beginning of the genetic era, I think, for neurodegeneration. And we need to follow essentially the cancer playbook, which is look at the central cell, the neuron, and all the cells around it, the glia, in some form of model system.
Great, thanks, Richard. Greg, maybe I think Mark, officially our session is over at 1:15, but I think you said it might be okay to stay on a bit longer, but maybe we can ask the others to do kind of rapid fire mode so we get a chance to hear from everybody before we close out. So
yes, for you, Robert, that's fine. Yeah. So, but please, yeah, whatever is appropriate, but soon.
Okay.
Thank you.
Okay, so I think if I had to pick one element of dogma that I think we really need to move farther away from, I think there's movement away from this, but it's not quite where it needs to be yet. It's thinking of clinical presentation or clinical syndrome as equivalent to disease. I think that is a real slippery slope in neuroscience and in neurology, given the clinical pathologic heterogeneity in many of these disorders. And that has been a real lesson learned the hard way in therapeutics development, right? So I think that flows into a potential opportunity. I think, echoing what many have said, I think there's a real potential for enhanced academic industry regulatory partnering to come together to identify those subtypes of disease, identify those multiple mechanisms at play in a given phenotype, design, more accurate, more sensitive clinical trials with better endpoints, and then have that meet the regulatory standard to bring new therapies to patients who need them. I think that is a core aspect of what we need to be doing and driving towards.
Excellent. Linda, maybe I can go to you next.
Yes, I think one particular dogma that I think would be interesting to kind of move away from is the idea that we have to actually do things in model systems before we test in humans, because ultimately the human case is really what we're trying to help. And I think as we develop therapeutics, we really should design within these trials these kind of window of opportunity type designs where we actually take brain tissue or take CSF or blood or whatever it is from the treated patients and analyze them, because I do think that sometimes our treatments cause an effect. For instance, if you give a tumor vaccine, you actually cause this influx of immunosuppressive cells that actually go and fight off your therapeutics. So I think having well-designed window of opportunity, tissue collection and strategies would be very helpful, because as Viviana pointed out, sometimes even though you have a good treatment, we just can't get it into the target cells that it needs to get to. And I've been approached many times by people who have found something in the lab and it's like, well, can you put it into the basal ganglia? And I was like, I can't do that in a safe way in patients. So that would be what I would say. I think, better collaborations among, everybody involved so that we could design these to really meet the needs of the patients.
Thanks. Viviana?
Two notes, one on dogma and one on heterogeneity. So one way to try to help with the dogma problem that was discussed was to not look in the silos of one disease at a time, because what are those besides names? There's a lot of heterogeneity within the disease, and then there's commonalities if we go from Alzheimer to Parkinson. So where preclinical or early stage work and trainees can help is by looking at this from a fundamental molecular cellular genetic perspective and find commonalities that cut across neurodegeneration, for example, such as lysosomal storage enzymes, or fundamentals that can help when acted upon a large array of indications. And by doing that in a way that's not necessarily mutation specific or disease specific or patient specific. And this is where the new generation of technologies that can act in an agnostic way as a toolkit, but when intersected with the patient, it can create a very personalized solution. And I will just give one example that has to do with CRISPR activation or CRISPR inactivation, for example. If you have two copies of the gene, if you have a good one and a bad one, you want to downregulate the bad one, you want to upregulate the good one, and you can do this with methods such as CRISPR-A and CRISPR-I in a mutation agnostic way, because it can go there and across the patients that are included, fix this in a more generalizable way. Of course, the technology is early. We have things to figure out on that, but trying to develop a robust toolkit that will, when intersected with the patient, can create these personalized solutions. So I'll pause there so Dan can close us off.
Yeah, they're really beautiful statements. I really agree with everything. I would just add that, maybe one dogma that I can add is this notion of disease. These things that we're talking about, Parkinson's, Alzheimer's, so far, autism, schizophrenia, other things that haven't been mentioned, these aren't diseases in the way that tuberculosis is a disease. These are syndromes. There's been a kind of pathological, I think in neurology, which is where I'm based as a clinician, fallacy that, because we can see something, that they're unitary. And, I think, just like breast cancer isn't one disease, just like, et cetera. So I think that as we begin to discern the very distinct etiologies and mechanisms being helped by genetics, but that's not all. I think that will help us begin to reformulate our notion of what these, collection of diseases, disorders are. Clinically, it's important to, use these things. Don't get me wrong, but from a drug development standpoint, I think it's really essential to recognize that these really aren't diseases in the standard, infectious disease kind of formulation, Koch's postulates, et cetera, all of that. So,
great, thanks, Dan. So I think I'm going to try and wrap us up. I guess my bit of dogma, which I hope everybody has already dispensed with, is that a disease starts when you're symptomatic. These diseases start years before people actually are manifest. And really, I think getting into those pre-symptomatic prodromal phases is the only chance we have of treating them in the case of Huntington's when there actually is a striatum to treat, as opposed to once it's already gone. And the opportunity, I think, is, and this was stated in different ways by the panelists, is this opportunity to do experimental medicine. There's nothing more precious to a drug hunter than an observation that's made in the population they seek to treat. And those types of observations are really where the breakthroughs happen in terms of finding the types of intervention points, the types of mechanisms, the types of targets, the types of biomarkers that are really going to get us to modulating the human pathophysiology. So I'll stop there with a big thank you to all of my panelists. I think it was a great discussion. I was here for the pre-show and I still learned some stuff during the live discussion today. A big thank you to Mark and to TDG. It's a privilege to be a board member and to see this lovely event, come together. And a big thank you to our attendees for your attention. So I'll stop there and say goodbye.
Thank you, everyone. Thank you, Robert, for the excellent moderating job.